Hi there, this is Brendon Riley and Ryan Goodspeed from Biology 1306, Section 003, and we are doing our extra credit video over DNA. We'll start off with the history of DNA. In order to understand how DNA works, it is important to understand the history of DNA. In 1857, Gregor Mendel conducted an experiment using pea plants to understand genetics and hereditary factors. Ultimately, Mendel discovered that genes are located along chromosomes. This discovery is significant because it communicates inheritance or the transfer of genes and traits from one's parents. So how does this relate to DNA? Well, in 1928, Frederick Griffith was working on a project to prove that DNA was the molecule of Mendel's discovery, inheritance. Griffith's experiment involved injecting two types of pneumonia into mice. He injected a viral pneumonia into one mouse, and the mouse died. In the other mouse, he injected a non-viral pneumonia, and the mouse lived. Then Griffith heated up the viral pneumonia and injected it into another mouse. The mouse lived. Finally, he injected the non-viral pneumonia and viral pneumonia that had been heated up into another mouse. This mouse died. With this experiment, Griffith discovered the viral pneumonia had been heated up, passed on a trait to the non-viral pneumonia, making it deadly, which follows the inheritance discovery that Mendel founded. Passing on the inheritance molecule was what Griffith called transformation. 14 years later, a scientist named Oswald Avery took Griffith's experiment one step further. In his experiment, Avery destroyed the lipids, ribonucleic acids, carbohydrates, and proteins of the viral pneumonia. Transformation still occurred despite this, however. Then Avery destroyed the DNA, and transformation did not occur. This is when DNA was finally revealed as an inheritance molecule. To understand DNA better, scientists were trying to make a model to understand how it worked and what it does. Erwin Chargaff in 1947 took samples of DNA of different cells and found that the amount of adenine was almost equal to thymine, and that the amount of guanine was equal to the amount of cytosine. Therefore, the idea of A equals T and G equals C became Chargaff's rule. In 1951, Rosalind Franklin was the first ever to take a picture of DNA using an X-ray crystallograph. 2 years later, James Watson and Francis Crick saw this picture that shows that Rosalind Franklin took. They then created the first model of DNA that shows it as a double helix. This model was revolutionized the study of DNA.
The structure of DNA. So what does DNA structure look like? Each DNA molecule is made up of two long chains arranged in a double helix. Each chain linked is one of four kinds of chemical building blocks called nucleotides. The functions of DNA. The main function of DNA serves as a central database. When we begin life as a single cell, our DNA has been inherited from our parents. As cell division occurs, our DNA transmits copies of itself to our trillions of cells. For instance, the genes along the length of the DNA molecule encode the information for building the cell's other molecules. This way, DNA controls the development and maintenance of the entire organism and everything it does. Forensic science of DNA. The study of DNA and understanding DNA can be used in different occupations such as forensics. For this specific job, DNA can play a crucial role in solving crimes because it links those crimes to people. DNA can be obtained through either fingerprints or samples of hair, blood, or any other bodily fluids. Then the sample is analyzed and compared to other DNA profiles in its database. Therefore, understanding DNA has transformed over the years from a basic understanding of genetics to solving crimes. This site is our works cited page. If you would like to know more information on DNA or how DNA works in forensics, you may visit these sites. Thank you for your time and have an awesome DNA day.